Think of metabolomics as a way to eavesdrop on a cell's chemistry. It looks at small molecules, metabolites, usually under about 1,500 Da, that sit downstream of genes, RNA, and proteins, and change quickly with diet, environment, or disease. Because of that, metabolite profiles give you a very direct readout of phenotype and current biochemical activity. There are two big approaches. Untargeted metabolomics is like a broad survey. You try to detect as many features as possible to generate hypotheses or find new biomarkers. Targeted metabolomics, by contrast, focuses on a predefined panel of compounds and aims for more accurate quantitative measurements. For tools, mass spectrometry coupled with chromatography, LC-MS, GC-MS, CE-MS, is the workhorse for sensitivity and coverage, while NMR gives highly reproducible, quantitative data and structural clues. You'll also see imaging MS for spatial maps and direct infusion, shotgun MS when throughput matters. A typical study follows a familiar arc. Design the experiment and collect samples carefully. Think fasting, timing, storage. Extract metabolites in a way that suits the chemical classes you care about, and run your samples with quality control checks and internal standards. The data pipeline then involves peak detection, deconvolution, alignment, normalization, and batch effect correction. For analysis, you'll mix things like simple univariate tests, multivariate tools, PCA, PLSDA, and machine learning for classification or feature selection. Identifying metabolites is done with accurate mass, MS/MS spectra, retention indices, and, ideally, authentic standards. Finally, you interpret the results with pathway mapping, enrichment analysis, and by integrating other omics. It's not without challenges. Many detected signals remain unidentified, matrix effects and ion suppression complicate quantitation, and ionization efficiencies vary by compound. Good QC, internal standards, and following reporting standards e.g. the Metabolomics Standards Initiative help a lot. Sharing raw data and spectra in public repositories speeds up validation and annotation by the community. Applications span biomarker discovery, drug and toxicology work, nutrition and microbiome studies, plant biology, and systems biology. Hot areas to watch are single-cell and spatial metabolomics, AI-powered annotation, and deeper integration with genomics and proteomics to trace causal biochemical networks. Handy resources include HMDB, Metlin, MassBank, GNPS, KEGG, and lipid maps for annotation and pathway context.